Welcome to Circulation on the Run. I am your host, Brad Maron. I am the Editor-in-Chief, and I could not be more thrilled to be talking to you today from Boston, Massachusetts at the BCVS Council meeting, where I've had a great opportunity to meet and see incredible science, which we're all hoping forms the submission stream for circulation and the other AHA circulation family members. I also couldn't be more thrilled than to be here with Dr. Sean Wu. Sean, congratulations on becoming the new BCVS Council Chair. Maybe you could take a few moments to introduce yourself to Circulation Nation. Tell us a little bit about your background, and then we'll talk a little bit about BCVS.
Brad, thank you so much for having me here for the podcast. I am the Professor of Medicine and Pediatrics at Stanford University. I'm also the Senior Vice Chair for the Department of Medicine at Stanford, as well as Associate Director for the Cardiovascular Institute there. I have been actively involved with the AHA ever since I was a pre-doctoral student way back in earlier days in the 90s, and I've been really well supported from the AHA, both scientifically and also with the research funding over the years. So being part of the AHA Council and also now taking on the leadership for the BCVS Council help push forward the basic science and translational research community into exploring new ways to engage the opportunity in research and also to develop new drugs and discoveries in the future.
Absolutely. I've been here for the last several days. Extremely exciting environment. You can feel the energy around science. I'm told there are over 5,300 members of BCVS now. Incredibly storied history to this council and the number of great scientific contributions that have been made, not just in the classic cardiovascular diseases, but more broadly in research in terms of methods and other important discoveries. It's a lot of responsibility for you. Tell the audience a little bit about your vision for BCVS.
BCVS has a premise around the idea that we want to be very sort of science-driven, data-driven, and focus in understanding the mechanism of basic cardiovascular physiology, understanding the genes' functions and regulation, and also how the genes are involved in various different disease processes. So for the BCVS, in thinking about how we might be able to help make the best contribution into the science and the health of cardiovascular patients, we want our members to be able to understand how the entire spectrum from the early discovery leads eventually to therapy. So one of the area which we have previously sort of somewhat dabbled but not really more actively pursue is to be better connected with the industry. So one example for this meeting here at the BCVS session this year, we have a new session called the Bench to industry. So basically we invited people who have either experience with the industry or people who are currently in industry to come and talk to our BCVS member at this meeting about what are the process of discovering new drugs and how the basic science that people worked on eventually make its way all the way into a therapy. So in starting to think about how the entire spectrum from discovery to translation all the way to therapy is something that I'm very excited about pushing forward. There are other things that we think the BCVS Council could certainly be also more proactive. So one example is that we have had this very broad international presence from all the scientists coming in from all different countries. So one aspect of the way that BCVS has worked very well is that we have partners and colleagues in other countries, and we've been invited to the meetings that are happening in these other countries, and they have been very actively involved in the AHA meetings at the BCVS and at the November session. So we've been exploring ways for the BCVS to become better connected. But the third thing that I think is an opportunity that we have also, again, explore a little bit, but maybe not nearly to the extent that we could, is the cross-council collaboration. So BCVS being usually the largest council within AHA, have been, I think, somewhat inwardly focused of what to do in helping our members to achieve the science goal that they want, but we may not necessarily have done as much with other councils, but there are great opportunity to do that. So lots of our scientists are involved in things related to gene and genome and gene editing. We have a fantastic GPM council who also are very similarly minded. We want to connect better. So one example is we launched a new science committee called Genetic and Genomic Therapy, and that is a partnership with GPM. So examples like that are what I would love to see us continue to push forward into having more of these cross-council activities.
Well, that's all very exciting. So bench to business, broaden international collaborations, and cross-fertilizing science between different councils that are already in place at the AHA but may not be talking as readily to one another as they may should. Give us an example of cross-fertilization between the councils.
Yeah, so we have a lot of people interested in heart development and congenital heart disease. And one aspect that we would love to explore is, for example, with the Young Hearts Council, they, of course, are interested in disease in the young and congenital heart disease being a predominant one that they focus on. So we would love to find a way to incorporate people from Young Hearts to come to the meetings of BCVS.
Sean, this is a very exciting time to be in basic science. The proliferation of high throughput assays, for example, that now make accessibility to omics across so many different domains readily available at low cost has created a huge amount of data for scientists to sort through. When you're thinking a little bit about reading and interpreting basic science reports that are presenting this level of information, what are the techniques and methods that you see as being effective at finding the most salient and functionally relevant features to those big data sets?
I agree with you that with the explosion of technology that enables collecting and presenting big data, we are somewhat in this transition to figure out exactly how do we evaluate the quality of those big data, how do we extract information that's most useful from this big data, and it's certainly still an ongoing process to understand what is available to do that. I do think that the technologies that are coming forward, for example, having better machine learning and AI, will hopefully help us find out what exactly the big data is telling us. But along with that, we need to have more science around the quality and also science around the rigor of the research that are being presented from these large omics data. And I think we are probably still somewhat behind in having those quality and rigor sort of develop so that people can really understand when a new paper comes along and a lot of omics data is being shown, are they meeting what those criteria are for having big data presented? You know, people don't really know exactly what is the right N for those omics data. People don't really understand like what are p-value that ought to be, so the level of the rigor you would have for certain level of big data. So we really need to have people spend time exploring, thinking about those things so that we know what would be the appropriate guideline for omics type of data, as the way we have now much better information about what a cell culture assay or animal experiment ought to have in the paper.
Very interesting, and in some ways gets us to a more contemporary framework of understanding context to those findings. We're seeing more and more every day, it seems, reports that bring our attention to environmental or acquired determinants of disease. How do you think about the convergence point of genetic context and these acquired disease drivers? Is that a direction that scientists in BCVS should be thinking?
We have historically been very reductionist based. So whenever we try to do a study, in the need to be rigorous and the need to be reproducible, we tend to have to be very focused and narrow around certain things. So when we see an intervention or perturbation, it's going to have to be a single intervention or perturbation, say at the genetic level in a certain pathway in cells or an animal, or if we're doing intervention perturbation in, say, environment would have to be so one exposure, so exposure to smoke or exposure to toxins. But now knowing all the various different determinants at the multiple front, so multiple gene variants that influence disease, multiple environmental factors that are simultaneously interacting with one another, we do have to really think about how to deal with those interactions that exist. I think many efforts around correlating the genotype-phenotype or PheWAS study to try to understand all the exposure to a certain entity simultaneously is pushing us in the direction of being able to be better at understanding how these kinds of environmental factors interact with genetic factors. But that's still relatively small subset of all the research that's being done. I think because historically we're so reductionist minded, so it does take, I think, some time to really, get people to change the way they've been doing science, to start being much more broadly inclusive of the different exposures.
Yeah, so it sounds like experimental models may benefit from contemplating genetic or even germline risk factors with environmental perturbations. We could even take that one step further and think a little more about multi-organ system research. We saw just last month the AHA advanced the first guideline document on CKM. I think that's a stark reminder to us all about how the heart itself does not function in isolation. We're familiar, of course, with cardiorenal, cardiohepatic syndromes and important biological pathways that link those two organ systems together in cardiovascular disease. How should we be thinking about this in the context of clinical syndromes that cardiologists are confronting more and more on a daily basis?
I do agree that we have really underexplored these inter-organ interactions or multi-organ system diseases, which clearly is what a lot of the diseases are that affects more than just one organ. And I think because of that mindset of being reductionist, the scientists have not tried to do so much experimentally, even when they have the opportunity, say in the mouse model, when you perturb a gene, you clearly are affecting many different organs with perturbation of that gene, but because we're so reductionist minded, we focus on our favorite organ or our favorite pathway, and we only explore the finding from that particular organ or that pathway, and we completely ignore all the rest of the organ and what might be the consequences in that model system. So I do think it is a conceptual switch to get people to really start exploring all the other organs that are happening if you were going to do perturbation in a single gene or in a single pathway. But I do also think there are forces and constraints around that. For example, our funding is one that usually goes along with the particular organ. The entire NIH is based around institutes that are organ-based. So people need to, or at least feel like they need to focus on the finding of the organ for that institute to continue to get support. And again, also I think to some degree, many journals are organ-based journals. So when you are submitting your journal, you feel like, well, that journal is only going to be interested for the finding I have for that organ and not necessarily wanting to care how much other organs that I've also found that have the phenotype. So, I do think it's probably a community-based mindset change to be open to the possibility of how different organs interact.
Well, that's great. I think we're beating back reductionism here through your vision at BCVS, whether it be cross-fertilization scientifically or cross-council collaboration, the efforts that we're discussing today that may move beyond a single biochemical event to explain complicated diseases, both through environmental drivers, but also through a systems-based approach to research. Completely fascinating. Thank you for that input. I think now we'll move to the final segment of the podcast, which we call Get to Know the Guest. I know we saw each other not that long ago in Northern California. Beautiful, beautiful part of the country. Maybe you could tell us what you like to do if you're not in the lab on a weekend that takes advantage of that scenic and beauty of Northern California.
Yeah, so I started out during my undergrad years at Stanford. So to me, it has been about a 20-year cycle through the Southeast when I went to my med school at Duke and finished MD-PhD there and my residency in internal medicine. And then when I moved to Boston, I've learned and acquire a lot of appreciation for the culture and the opportunity for both people and science here in Boston. Now back to Northern California, I have managed to reconnect with a lot of the things that I have spent time doing. So during the winter, we as families certainly go up into the mountains at Lake Tahoe and enjoy the skiing. And I personally do snowboarding, so that's what I do for fun on the weekends during the winter. And of course, in the summertime, there's the beaches. So, we're very close to Santa Cruz. So, and also various different places along California coast, Carmel, Big Sur. So we try to take some of these weekend day trips around to see the beautiful scenery there.
Fantastic. So that's Big Sur to snowboarding, all within one space. Excellent. Want to thank you very much, Sean, for taking the time to talk to us, to talk to Circulation Nation. It's been a fantastic and very exciting conversation. I wish you the very best of luck, and hopefully our paths will cross again soon so we can update on how your progress has unfolded at BCVS. I'm sure with your leadership, they are in extremely, extremely sound hands.
All right. Thank you so much. I really appreciate doing this.